Okay, hi everybody. Good morning. Um, my pleasure to go on the last day. Thanks for sticking around. I get the opportunity to make jokes about all the other previous talks and put things in context a little bit, hopefully. If nothing else, I came up, I feel like, with a good title for this talk. We'll see if it lives up to it. Okay. Um, have found it curious, let's say mildly concerning, of the lack of memes this meeting. And I also find it funny that we haven't had so many U-map plots that we're staring at, maybe because Leo's here. But this is a good one I think we can all appreciate. So we just start off with a light joke, okay? I want to start with a couple of open questions because I really appreciated the direction of asking thought-provoking questions in this field. In my mind, maybe you have answers to these, but based on my conversations with a lot of you this week, I don't think we have good answers yet. But the couple questions driving this talk are, has single cell sequencing or analysis found the needle in the haystack, let's say specifically in cancer, where we think, okay, well, we want to find that single cell population, right? We want to find that rare cell type that's really driving what we're looking at. And then second question is, do single cells contain information about the system as a whole? And can we do something at the single cell level to integrate the information that all the cell and cell types are containing to understand how the system is evolving, let's say at a tissue or a whole body scale? So those are the kind of driving questions for this talk. And I want to start with, I love this slide because it's just a great story. When I was starting my first faculty position at City of Hope, this guy, Guido Marcucci, who's a physician scientist, was on my interview schedule. And I didn't know him. Okay, so I came into his office and he said, asked me this question. He said, in this really thick Italian accent, do you think we could write the equation of leukemia? And I wanted the job. So I was like, sure, why not? Let's try. And we've been working on it for like 11 years since. Okay. And I wish I could tell you a lot of the interesting things that we found over the 11 years, but I'm going to try to stick to the idea here of focusing on the questions at the single cell level that have come up. And a lot of them have come up by virtue of the fact that we decided early on in this project to view leukemia as a disease of the body as a whole. So leukemia is a blood cancer that originates in the bone marrow, and it evolves not only through its source in the home in the bone marrow, but through the system. It circulates through the blood. So especially 11 years ago, and I would argue even now, the dominant view of how to think about cancer evolution is from a clonal perspective. There's a malignant cell, it divides, it creates new mutations, it has daughter cells, there's a phylogenetic structure, but this view is very cancer cell centric. And what we wanted to do was look at the blood, the peripheral blood, and integrate the information that was contained in all the cells in the peripheral blood, because really leukemia as a unique disease in cancer is evolving and engaging the immune system in all kinds of different organs. And I took this, the slide out I usually put after this, which is Sui Wang, Professor Wang's paper about the end of the genetic paradigm in cancer, which I find very, very thought-provoking. And that is thinking about, instead of DNA mutations and single cell clonal structure, really about the evolution of the system as a whole and as a state transition. And this is doesn't really need to be explained to this audience after this week because I've talked a lot about potential energy landscapes. But let's say at a very high level, we started to think about just the concept that your body in a healthy state is in a stable equilibrium of hematopoiesis, okay? And that the probability of transitioning into a leukemic state is very low, and just conceptually illustrated by a high energy barrier between those two states. And that in the presence of any number of things, again, de-emphasizing specific DNA mutations, but let's say immunosuppression, chemotherapy, any number of things that can open up an opportunity for malignant cells to start to evolve, or your system to start transitioning into another state, reduces this energy barrier and makes the transition into, let's say, a leukemic state more likely. And because this is an incredibly simple conceptual mathematical model, we'd think about it in terms of these critical points, C1, C2, and C3, where really the transition state in this concept, especially given Herbie's talk just now, is not about a stable alternative state. It really is a transition state in which the system does not stay, and you're very unlikely to observe it in that state. So, the idea that as your body and as your overall immune system and bone marrow hematopoietic system evolves, it undergoes this critical transition to leukemia. And so we spent a lot of time developing mathematical models of this, which are of very little novelty and interest to this crowd, but to other crowds, they are more interesting. Thinking about a Langevin equation and that kind of potential where we model individual transcriptomes as going from a health state to a leukemic state, crossing this energy barrier. And we really spent a lot of time studying that with experimental data. Okay, so one of the things that I wanted to mention on this slide is that what ended up happening from Guido's question to me was this really rich interdisciplinary collaboration that's gone on for a long time. Okay, biologists, clinicians, physicists. I'm the token mathematician. Okay, there's a few others with me, but I'm the one that's in charge of dealing with the math aspects of this. But this really has been quite an amazing interdisciplinary sustained collaboration. And it was started by this experiment that Guido did. So when I told Guido, he said, "Can we write the equation of leukemia?" I said, "Yeah, sure, why not?" Okay, by the time I got to City of Hope, he had already started this experiment, which I'm embarrassed to even tell you how much this cost like 12 years ago, okay? But this is an inducible mouse model of leukemia. And the reason why it's important that it's an inducible model is because those of you who don't know, there's kind of two ways, let's say, in a very simplistic view of studying cancer in mice. One way is to just give the mice cancer cells, just inject cancer cells in there, and voila, they have cancer now. And then another way is to spend probably years and part of your postdoc or early faculty career developing mouse models that don't act that way. And this is one of them. So it's an inducible model where this mouse expresses this fusion protein, CM, that once it's induced, the mice organically, let's say, develop leukemia over time. And so we have controls that have the transgene, but it's not activated. And then we have leukemic mice that have the transgene activated with this poly-IC, and then we follow them, the peripheral blood, over time as they develop leukemia, okay? And in the peripheral blood, we started with bulk sequencing. I'm getting to the single cell part here, okay? But we started with bulk sequencing. So initially we did RNA, microRNA, and DNA methylation, and you see the survival graph over here on the right. You notice the x-axis of this graph? This was a year-long experiment, okay? This is a really, really long experiment. We did it many times. If there's nothing else that came out of this 10 years of work, it's that this experiment these observations are incredibly reproducible, okay? That take a long time to do. So we follow the evolution of the disease through the peripheral blood. And what we ended up finding very early on was that we could create a bulk transcriptome state space, okay? Basically with just PCA. And I'm also a little bit surprised by the lack of debate or passion or whatever word comes to your mind about dimension reduction methods besides UMAP around in this week, because they're so critical for understanding single cell data and genomic data. I have a lot to say and I have a lot of opinions of why I think PCA is one of the best, if not the best way to approach most of these kinds of analyses. But let's say what we did is we took all the time series data. And when you do PCA on it, you get PC1, PC2 here, you get these kind of two orthogonal states, a health state and a leukemic state. And the health state is indicated by these black dots. So these are the control mice that did not develop leukemia. And the red dots that are kind of pseudo-colored go from black down to bright red are the mice that ended up developing leukemia. And what was nice about this is not only are the principal components orthogonal by construction, but let's say now the states, the phenotypic states are also orthogonal. And this allowed us to then look at, let's say, the right singular vectors of the singular value decomposition and start to associate genes and gene weights with different states, okay, with the state of the leukemia as it evolved. And again, there's a lot to say about this, and if anybody wants to really get into it over lunch, our last part of today, then I'm happy to. I don't want to spend too much time on this, but one of the questions that we've asked ourselves and we've been asked for like 11 years is why PCA and which component captures leukemia in PCA. And I wish I could tell you why it's PC2, but it is, okay? PC1 is kind of correlated with time, and this is capturing a lot of technical variability as well as just, we think, kind of an aspect of aging. But if you just look at these first four components, PC2 just rings out like a bell. It's like the way I described it is like tuning a radio. Okay. The black are the control mice and the red are the leukemic mice. It's just extremely clear that that component captures the disease evolution. It just does. And for a long time, we wondered what PC4 was showing. And I was a little perplexed by that until I found this paper in PNAS by a very angry person. And he's very angry at PCA use in time series and space data. He's most angry at the neuroscience community, okay, for interpreting oscillations in PCA based on neural activity. But it was one of these papers that really kept me up at night because I thought, oh shit, I have to retract my paper. I've done something terribly wrong. And, but I've convinced myself since that it is not the case because one, we don't see any oscillations in our disease trajectories. Although there's there's structure, temporal correlations in our data because of the time series. But this is what explains this PC4, why there's this oscillation across all the mice and all the time points. It's really, really very fascinating paper if anybody's interested. Yeah.
What's the rows and columns of the matrix you did PCA on?
Genes and time points.
Okay. And all the samples are just concatenated that way?
Yes.
I'm just trying to wrap my mind around a PCA where there's like no negative values at all. Is something like for both PC2 and PC1 so can you just tell me what exactly is plotted here? Like what's
no negative values? Oh, okay. I'm sorry. The x-axis. Okay. Thank you. Thanks for asking this question. X-axis is time.
Okay, good. That makes more sense.
The y-axis is that component.
Okay. And so still, I see. So you do have some variation there. It's just a very small on one side, essentially.
Yes.
Okay, cool. Thank you. That makes so much more sense.
No problem. Sorry about that.
So when you do the PCA, did you use the time component or not?
We did not add time as a vector, no.
They were just separate samples, basically, all considered.
Yep.
And then you like connected them here using time.
Right, and then we connect them. Yes, exactly.
So, so you say every time point is a sample. You don't have many mouse, and every mouse is a sample, right?
So we have, in this case, there are 9 red mice that develop leukemia, and there are 6 control mice in black, and each mouse has approximately 10 time points.
Every mouse has 10 time points.
Yes. So we're following the mice over time.
Okay. And sorry, one more. The control mouse, are they wild type or are they non-induced?
They carry the transgene, but they are not induced.
You just say if I ever created a PCA like this, I would also always want to make PCAs.
Yeah. Well, it's a mouse model, so it's very nice and reproducible and consistent. So is it always going to work this way? No. But I will tell you it's worked in very many mouse models. Okay. We're up to like 5 different mouse models and it works for some reason. Okay. Very consistently.
PC4 you said was related to neural? Is it oscillations? Because
sorry, this is my speaking style, which I guess would be a little confusing, a little rambling. It has nothing to do with neural. It has to do with a paper that I'm not showing where the author was mad at the neuroscience community for using PC on time series data.
All right, okay.
And what he's mad about is a neuroscientist trying to interpret that oscillation as a neurological pattern.
Oh, okay.
But it is in fact not. It is a phantom oscillation. It's not there. It's not in the data. It's nothing real. It's not interpretable.
Got it.
Yeah.
Thank you.
Okay. And then what we observed also very quickly was that this transition is really global across the genome. Okay. So what you're looking at here is a phenogram, which is a karyogram of the differentially expressed genes that are either up or down at the first time point, at the transition state, and then at the final time point. And what I want you to observe from this is that there is differentially expressed genes in every chromosome, in every arm of every chromosome, both up and down everywhere across the genome by the final disease state. There are so many differentially expressed genes when you compare the leukemic state to the healthy state that it's uninterpretable. Okay, 16,000 differentially expressed, or 16,000 sequenced genes, 11,000 of them were differentially expressed. Okay, so like good luck making sense of that from any kind of pathway point of view. But that was all bulk. Okay, and this meeting is about single cell states. So what about single cell states? we've had a few grants on this topic, and every reviewer said, well, what about single cell? So, okay, fine, we had to do it. And I asked AI to, another thing there's not a lot of in this meeting is AI pictures, okay? I asked AI to make this picture of scientists looking for a needle in the haystack, and I'm actually, I kind of like it.
Where are you?
Einstein makes an appearance, which I didn't ask for. But I actually kind of like it. It's pretty, I don't know, interesting.
No, I don't like it because the search strategies should only have heuristics.
Yeah. Where's the magnet? Seriously. Okay. Yeah, right. I know. Yeah. Day 17, still looking. Okay, so what happened when we did a single cell study? Okay, so this is what we're here to talk about is do the same study, but longitudinally in single cells. So now we take the mice, do peripheral blood, longitudinal time points, single cell at every time point. Again, an experiment. I'm not going to tell you how much it cost. But we're finally getting that paper published. It should be it is online as of like this week, which was a coincidence. But what we found and what I am getting from a lot of the discussions that we've had this week is maybe not surprising to you, is that almost all the variance is driven by cell type. Okay. So what we did is we took all those time points, all those single cell data, all the data, and we did PCA, because that's my favorite. And PC1, PC2 completely driven by cell type. Meaning that the difference between T cells and B cells, stem cells and myeloid cells, et cetera, is greater than the difference between having leukemia and not having leukemia. I was kind of shocked, maybe not too surprised, but shocked to find that out. Okay. And when you color that same plot by time point, maybe you see a little bit of a purple, area right here that's coming up at the final time point. But the truth is that almost all the variation is driven by variation within and across cell type. Okay. And this reminds me of one of my favorite quotes, which I tell my kids and my kids' friends all the time. You're different just like everybody else. Okay. And that is kind of, I think, the metaphor for what I'm finding in single cell data is that the difference between cells is vast when you're looking at it from a point of view of 15,000 attributes or whatever it is, however many genes you have sequenced. Okay. And what we found particularly revealing is that the disease trajectories were only revealed after pseudobulking. Okay. So once you pseudobulk all the samples and all the cells and all the time points in black, what you see are these nice, beautiful disease trajectories, just like we saw in the previous. Okay. That totally matched our prior bulk. And that makes sense because it is like pseudo bulk. Okay. But once we bulked it and plotted it versus time, now we could start to see disease trajectories and we could see the difference between disease states.
How many mice did you have?
How
many mice did you have? Just out of curiosity.
So these are 3 CML mice, and then we did another 3 with a MIR142 knockout, which is like a blast crisis mice. So 6 and then controls. So 12 total, but this is showing 3 just for this point.
And the pseudobulking is basically by cell type here, right?
No, so on the left, what you see is the black, that's total pseudobulk. All cell types, all genes.
By mouse, or what does it mean?
By mouse. So each
By
one of
mouse.
those trajectories is a mouse trajectory.
Okay. So that basically says the single cell equals the bulk.
Well, kind of, and actually that's my next slide.
Oh,
so then what we do is pseudobulk by cell type.
Yeah.
So what happens when you pull all the B cells together, all the T cells, all the myeloid, all the stem cells, you start to now also see these kind of very similar but different disease trajectories. And this is not equivalent to the cell population frequency over time, which you see on the right. Sweet.
I don't understand the title. So how can you have a trajectory without pseudobulking? What would be the alternative?
Fair. Okay, fair. I guess the point I'm trying to make with that title is that we were not able to see a disease state by looking at the single cell level. Put it that way. Yeah.
So when you pseudobulk, how many cells are you averaging over typically?
Well, it's not an average, it's just a sum.
Okay.
But you mean in a per sample?
Yeah.
Per sample per time point? I think it's 20,000,
Okay.
20 to 50,000.
There's no way you have 20 to 50,000 T cells in one mouse.
No. No.
Yeah.
So no, pseudobulk total in the sample.
Okay.
And then it's broken down by cell type on the right.
So probably varies.
Is that what you were asking?
Oh,
Yeah.
okay.
I don't have numbers off the top of my head or in this slide about how many B cells there were in each sample,
Yeah.
how many, et cetera. Okay.
Oh,
Maybe be slightly off topic, but for people, in the old days of bulk RNA, one of the issues was people tried to derive methodologies for separating out cell types. Right, based on
Deconvolution.
transcription.
Right.
Can you go backwards and now use pseudobulking of real single cell data to see whether those algorithms actually work?
We could. The data is available. Data is available if you want to do it, Herb.
Well, it's how well those work.
Yeah.
Something, but
Yeah.
what?
They don't.
They don't. Okay.
I mean, mathematically, it's hard to believe it would. Yeah.
I bet you could. Yeah, we can talk.
Okay. Well, yeah, I'm running out of time, so I just have a couple more slides. So then what we did, and I think this is related to the question, is so then could we use some kind of information like technique, compute KL divergence of the cell type pseudo bulk and compare it to the overall bulk to kind of quantify the amount of information about the overall system carried by each cell type. And when we did that, we saw that, B cells and myeloid cells contained a lot of information, but that surprisingly, the stem cells contain the least amount of information, which the reason why that's surprising is because leukemia kind of is originating from stem cells. And there's two reasons for that. One is because the stem cell state was very difficult for us to parse and define, and they were more rare in the peripheral blood. So that might be, there's probably a reason for that. But we were able to kind of quantify the amount of information carried by cell types about the overall system. And then this is the last slide. And then what we did collaborative work with Adam and a postdoc in his group was to do some modeling to ask the question, okay, could multiple different cell type dynamics result in the same system level dynamic? So if the B cells were bi or tri-stable, T cells, myeloid, stem cells, all had different kinds of multi-stability in them, could they combine together to provide similar kind of overall picture of the system state? And that's a whole other analysis I'm not going to go into, but the short answer is surprisingly yes, using a mathematical model. Okay. So final AI here, which I find this funny because it's actually surprisingly kind of coherent. Missing the forest for the trees and single cells and tissue is what I asked it to draw. But the inset on the forest is the bark, and the inset on the biology is the tissue, which is like mixed up, right? So anyway, close, but not quite. So then the open questions in my mind, I think, based on a lot of this work is, has single cell sequencing and analysis found the needle in the haystack in cancer? I would say no. Okay, just based on the studies that we've done and based on what I'm familiar with. But I mean, I'm open to argument on that. But I think that what we can do is take our single cell level data and combine it in different kinds of ways, maybe to get a better picture of the system as a whole. And part of what Peter Sims was talking about was overall tissue state and integration of multiple cell types within a tissue. I personally think that's kind of the way to go in terms of viewing the disease as a system-wide transition.
Russell, can I ask you a more philosophical question about needle in the haystack?
Yeah, because this is it. I'm done. Yeah.
Is it because we don't even know what needle looks like in single cell? Because it's not a true analogy, right? Because in the needle in the haystack, I know what a needle is and how it looks like, which is not the case with a lot of things. So is it also like this definition of what you're looking for?
Yeah, I mean, it could be that. I guess the idea in leukemia is that there's this very specific cell type that's going to precede the disease, and that you would see that there, and then you would see it persist, and you wouldn't see it in mice or systems that didn't have the disease. That would be the kind of needle in the haystack, the cell type that's preceding the disease state. And I don't know how best to define that other than its presence.
That it has to be there.
But it could be a classification problem. And I mean, part of what we've talked a lot about this week is cell identity. How do you even define that in this huge continuous spectrum? Maybe that's one of the challenges.
Yeah. I guess just to that point, I mean, like leukemia in particular, I mean, there might be just a vanishingly small population that contributed to all these, right? So actually, this is like maybe the most difficult needle in a haystack problem to attack with this, right?
That's a possibility. The reason why I find that unlikely, let's say in the mice, and so one of the other big kind of caveats here is that this is all mice. Okay, so this you can post this on Twitter, the in mice account. Okay, but we've also looked in the bone marrow, and we don't see those populations emerging in the bone marrow in these systems either. Bone marrow is harder to longitudinally sample, but I mean we've done SAC experiments with that.
Thank you. Maybe I misunderstood something. I have I don't know if this is a technical or a terribly stupid question. Like when you were showing these trajectories, now I saw the compositional effects of the different cell types.
Yeah.
I wonder because you didn't show any genes or any of the loadings of the principal components that carry the signal. And often when I've done pseudobulking in the past, I have to be very cautious with pseudobulking because the aggregating noise can carry information of maybe the responsible cell type. For instance, like imagine that you look at the principal components and all you see as MS41 and B cell genes, or who knows. Like, have you looked at that? Like, see if the genes make sense and they seem to be not mixing the signal across the populations and seem to be driving your whole system.
Yes.
And none of that happens.
It's not that none of it happens, it's that it's very difficult to interpret. So, yeah.
Let's continue questions. Thank you very much, Russell.
Yep.
Let's continue questions over coffee.